Good morning everyone. Welcome back to Biology with a Difference. Yesterday we talked of incomplete dominance, where one allele was not completely able to dominate the other allele in a gene, and hence we had different results from what Mendel has given us. Today we will study codominance, where two alleles will express themselves separately. We have two examples in this case. One is color in cattles and the other is blood group AB. First we will discuss the coat color in cattles. Coat color in cattles. You might have seen cattles of different colors, that is white, brown, black, or sometimes you might have seen patches in the color or skin of the cattle. That is what we will discuss here, the cattles who have patches on their skin. The gene for coat color in cattle is capital B, responsible for black color, and small b responsible for white color. So according to what we have studied, this should be dominant, but this is capital B, only one type of gene because it is homozygous. This is small, that is gamete, because it is homozygous, and they will result into capital B, small b. So all will be of this type. What happens, this B is able to form coat color which is black in color, and this B is able to form coat color which is white in color. So the animal will have both the coat colors. That means it will have somewhere patches of black, somewhere patches of white. So both black and white are able to express when they are in heterozygous condition. So this is also expressing, capital B is also able to express. We have black color over here and white is also able to express. So in a single organism we have both the black and white color when the gene is in heterozygous condition. So this type of dominance is called codominance, where both the alleles are able to express in the organism. So this is an example of codominance. Let us see another example in human beings, that is blood group AB. I am only talking of blood group AB. The other blood groups that we have, we will discuss later when we will study multiple allylism. So blood group AB, this is an example of multiple allylism. That means it has more than two alleles in a gene, but in an organism there will be only two alleles to form a gene. So what are the three alleles in this case? It is IA, IB and IO. These two are considered to be dominant. That is, IA and IB is dominant, whereas IO is recessive. So what will happen? If we have IA and IO, we will have blood group as A. If we have IA and IB and IO, we will have blood group B. That means A is dominant over here and B is dominant in this case. So both these alleles are dominant when present with IO. But what will happen if both of them are present simultaneously, IA and IB? What is the role of this IA? This IA produces antigen A on RBC, that is a protein on RBC. So antigen A is produced on RBC. Similarly, this IB will produce antigen B. We write antigen in capital. So antigen A is produced by IA and antigen B will be produced with IB. So this is now when we have both of them together in a gene, that is we have IA and IB, both of them are together. So what will happen on RBC, we will have both the types of antigen, that is antigen A and antigen B. So both of them are able to express when they are present together. This is an example of codominance, that where both the alleles are capable of expressing themselves. So blood group AB is another example of codominance. Next is multiple allylism. Just now we will discuss this blood group in details now. ABO blood group in humans. ABO blood group in human is an example of multiple allylism. Multiple allylism means that it has more than 2 alleles in an organism. Whatever we have studied till now is that all of them were having only 2 alleles. That is, if we had that we had, let us take example of tall and dwarf. What were the 2 alleles? One was capital B and other was small b, and we could have combinations, 3 combinations possible with this help of this, and that was capital B, capital B, capital B, small b and small b, small b. So whatever we have studied till now, we had only 2 alleles in that gene or for that character. But now what we are going to study, we have let me write it once again, that got erased by mistake. So in case of human being, we have multiple allylism, that means more than 2 alleles, IA, IB and IO. Just now I discussed this. These 2 are dominant alleles and this is the recessive one. Now what will happen? We can have, but one organism will have only 2 alleles in a gene. So what will happen because of this? We will have many combinations because of this. Let me IA, IO, IA, IA, IB, IO or IB, IB. Another combination is IO, IO, IA and IB. So this will show which blood group, because A is dominant, we have blood group A. Here we have both of them as homozygous, so the blood group A. Here we have heterozygous, but B is dominant, so blood group B. Here we have homozygous condition, so blood group B. This will have blood group O, because none of the dominant are present, 2 recessive together will have blood group O, and this will have, we have studied, both of them are able to express themselves, so blood group AB. If we look at phenotype, that means what are the different types of blood group present in human beings. We have how many blood groups? 1, 2, 3 and 4. So, we have 4 blood groups in human being. And if we look at genotype, how many are there? 1, 2, 3, 4, 5 and 6. So, 6 genotypes are there for 4 blood group. So, the phenotype is only 4 and we have 6 genotypes for blood groups that we have in case of human beings. This is homozygous, this is also homozygous, 3 homozygous conditions are there and 3 heterozygous conditions are there. IA, IO is heterozygous, IB, IO is again heterozygous, IA and IB is also heterozygous, and 3 homozygous are IA, IA, IB, IB and IO, IO. So, but there are only 4 blood groups. So, phenotype is 4 and genotype is 6. This blood group, ABO blood group in human beings is an example of multiple allylism. Can we find out the parent if blood group is given? Suppose father has blood group A and mother has blood group B. If in a question it is written that father has blood group A, we have to take heterozygous condition. Until and unless it is mentioned that it is a homozygous condition, we will take it as heterozygous. So, what is the genotype for heterozygous? IA and IO. Genotype for homozygous? IB and IO. This is the genotype. No, because it is heterozygous, 2 gametes will be formed, IA and IO. Here also we will have 2, IB and IO. If we draw a Punnett, I always draw it. So, it is IA, father was IA, IO, mother is IB, IO. So, what will be the genotype for this one? IA, IB, IB, IO, IA, IO, IO and IO. So, this will express IA, IB will express which blood group? AB. IB, IO will express which blood group? B. IA, IO will express which blood group? A. IO and IO will express which blood group? O. So, we have 4 out of 4, we have 1 for each of them. So, every blood group is present in the population, 25% chances are there of each blood group. That means, if a child is born, he may have, there is 1/4 chance that he may have AB or B or A or O. But if we change this heterozygous condition to homozygous, let us see what will happen. IA, IB. Only one type of allele because we have homozygous condition. Here also only one type of allele because we have homozygous condition. So, all the parents will be of AB type. 100% offspring will have blood group AB. So it is possible to find out the phenotype of the children if we know the genotype of the mother, or it is possible even to find out the, if we know the different children what are they are having, we can find out the father or mother's genotype. So this is all for today. Hope you had a great learning. If you liked it, share it with your friends, and don't forget to subscribe and click on the bell icon to get the notification whenever a video is released. Thank you once again.